I'm going to share my screen. Okay. So we're working in this BISB, or not BISB, the Biomedical Research Facility, which is in the medical school. And I'm going to talk about all sorts of enormous projects. And so one of the reasons that my laboratory uses a lot of bioinformatics is that we really do a lot of big data stuff. You know, we're not sitting there by ourselves, you know, trying to figure out how to make a new molecule. We're doing big things with, and in order to do big things, you have to collaborate with all sorts of people all over the world. So we're a little bit more in terms of, you know, in the nuclear physicist mode of things, not quite, but that's a little bit more the way that we work. Anyway, so I'm going to be talking about this story, and you know, this is work that's happened over the last 15 or 20 years, but it sort of tells you how, you know, we made a new drug for malaria, and you know, how we used bio and cheminformatics in the process. So, so even though we hear all about COVID-19, malaria remains one of the leading infectious diseases. And so, you know, it's really confined to sub-Saharan Africa and parts of Asia, but within sub-Saharan Africa, you know, I think there were 200 million cases in 2017 and about 500,000 deaths. And this is something that comes back and hits you every year. So, you know, people who are living in sub-Saharan Africa, you know, have to deal with malaria repeatedly. So it's spread by infected mosquitoes. And it has such, it's actually more deadly than COVID-19. So I think the death rate for COVID-19 is, you know, 2% or something like that. Now, I think that's probably similar to what malaria would be if you weren't treated. So there are good drugs to treat malaria. So that's the big difference. So with COVID-19, we don't have anything that's a really good drug. But with malaria, we do have good drugs to treat it. But the reason people die and children die from malaria is because they don't get to the hospital in time, which is also possibly the reason people die from COVID-19 is that, you know, that you get short on breath and you don't get to the hospital in time either. And but anyway, it's had a really big impact on the human genome with, you know, I think the probability of a child before, you know, even up to a few years, 10 years ago, the probability that you would die before your 5th birthday from malaria, if you were living in some of the, you know, remote area, not so remote, but poor areas of Africa would be like 20%. So, you know, out of every 5 kindergartners that you could have, one of them would have probably died from malaria before they reached their 5th birthday. So even today, it remains a really, really big problem, but we don't hear about it because it affects poor people, often people that are trying to survive on $1 or $2 a day. So it is spread by mosquitoes, and it's a parasitic infection. So, and you may have heard of multiple discussions about, you know, human malaria being caused by different types of parasites, and the most lethal one is Plasmodium falciparum, which is found primarily in sub-Saharan Africa. Another type of Plasmodium is found more in Asia and India, and that's Plasmodium vivax. It's not quite as lethal. So the nasty thing about malaria is that you can get it over and over again. Can you guys see my slides, right?
Yeah, we are.
Okay. Cat just knocked or something. Okay, so you can get it over and over again, and the severity of the symptoms decreases with time. And you know, the worst cases are going to be when you're, you know, a little kid, unless you're, of course, not from Africa, because then you can get it. And sorry. No, my cat just did something naughty. Never mind. anyway, so you get it, you can get it over and over again, and the severity of the symptoms decreases with time. So you never actually can become immune to malaria. It makes you a lot less sick after you've had it multiple times, but you don't really get immune, because the parasites that cause the malaria are always changing the antigens that are expressed. And just to tell you about the life cycle. So the mosquito will bite you and it will release these sporozoites into your bloodstream. And these will go to your liver and they will set up a quiet infection in your liver that can last up to 10 days. And during that time, you feel fine. You don't know anything that's going on. And then after about 10 days, the malaria will break out of your liver and start infecting your red cells. And when that starts happening, you get a massive immune response. It's sort of like the same sort of symptoms that you would get with COVID-19. You have inflammation and you have anemia that comes from the parasites lysing your red cell. Ultimately, some of these, the infected red cells will get into your brain and it will cause inflammation in your brain, and then it will push on your brain stem, and then the kids will basically just stop breathing, which is really how malaria ultimately kills you. So that's the bad news. And the good news, and there's no vaccine, no one's ever been able to make a malaria vaccine. So the good news is that there's a variety of different drug treatments that have been available for many years. There's some old Chinese herbal remedies that, from which we have a natural product that works pretty good, and those are artemisinin and combination therapies. I think they've been known that the extracts from this plant were able to reduce fevers for about 2,000 years. Isolating the active ingredient over the last 20 or 30 years resulted in the Nobel Prize for Tuyuyu. It's one of the first women to get one of the few women to actually get a Nobel Prize. And I think, was she one of the first people from China as well, I believe. So the problem with malaria is not that you can't find things to kill parasites, but resistance becomes a problem. And the parasites that are out there these days are resistant to most of the treatments that are available. So does anyone have any questions at this point?
Question, just raise your hand. I think I'll ask to unmute.
Wait, I have one. Why is malaria different than other diseases in terms of how you can catch it multiple times?
Okay, so the parasites that cause malaria are fairly sophisticated. So they're eukaryotic pathogens. So, they have about as many genes as, for example, Saccharomyces cerevisiae. They're not bacteria. And they have developed a very unique system of changing the expression of genes on the red cells. So you make antibodies that attack infected red cells, but the parasites will rearrange the genes so that you're now making a new set of antigens. So it's the same way that you get the flu over and over again, right? Because the flu that you get this year is different than the flu that you got last year, because a lot of genetic variation has happened. And that's ultimately the same reason that you get malaria over and over again, is because the parasites are switching the antigens that they have. And in most cases where you have a disease where you get really good protective immunity, you get sterilizing immunity like polio, where you have really effective vaccines, there isn't nearly as much shuffling of the genome from season to season. Does that make sense?
Yeah, thank you.
Okay. Next slide.
I had one more question.
Sure. Sorry, I'm not seeing the hands up anywhere. So. Oh yeah, okay, Gina, okay.
Yes, I was wondering what impact the parasites had on the mosquitoes.
Well, we don't think they have much impact, but that's a really good question. We keep thinking there must be some reason that the mosquito hasn't lost the genes to get rid of it, because it seems like, you know, having parasites usually isn't a good thing. On the other hand, you know, I mean, we all think of terrible stunting that comes from having worms and so forth. But on the other hand, you know, having worms seems to make you less susceptible to autoimmune disease. So people who have often had a lot of parasites are often a little bit less susceptible to allergies and things like that. So, but we don't know, you know, why you know, if there's some fitness advantage that's conferred to the mosquito if it gets infected with malaria, but it seems to you know, to really have developed a very good symbiotic relationship between the mosquito and the parasite. In parts of Uganda, you might get 100 infectious bites a night from mosquitoes.
Sorry, I also have a question. I was wondering, is it true that being like heterozygous for sickle cell anemia is like a kind of, I guess, pseudo immunity to malaria? Because I've heard that before.
That's true. And so the sickle cell anemia affects the parasite's ability to replicate in your red cells. So if you're a heterozygote, you have a good protection against cerebral malaria. And that's why sickle cell alleles haven't disappeared, because the homozygous is very, very lethal. I think you die right away if you're homozygous for sickle cell. But if you're heterozygous, you're protected against malaria, which has driven the sickle cell allele to higher frequencies than you expect in Africa because of its protection. In addition, you know, there are other hemoglobinopathies that provide protection from malaria that are found in other populations that are exposed to malaria. So, you know, the data is that malaria is probably the one pathogen that has had the biggest impact on the human genome because of the high death rate that it causes. Up there with the plague and cholera probably as well. So actually TB and malaria probably number 1 and 2 in terms of, you know, impact on the genome and humans developing protective, you know, evolving protective alleles. Any other questions? Okay. All right, next. So, oops, sorry. Number of cases of malaria were going down for a long time. Well, actually, this has gone up and down over time. You know, malaria was a really big problem, probably kept a lot of people from going to other countries, you know, 500 years ago, because honestly, if you didn't have any protective alleles and you even went, if you were from Northern Europe and you went to Italy, you know, they had a lot of Italy and Rome. And, you know, they would say, well, you had to be an Italian pope because you would die if you were from England and then you went to Rome and then you were bitten, you know, exposed to malaria. And it also probably kept people from exploring parts of sub-Saharan Africa, you know, just because, you know, it was 500 years ago, it's really hard to keep yourself from getting bitten by mosquitoes. You know, we know it's hard enough these days to keep yourself from being bitten, but back then where we didn't have any nets or glass in the window and your house was full of cracks, there's probably a much higher chance you got bitten by mosquitoes. So anyway, the number of cases have gone up and have gone down. They discovered chloroquine about 500 years, or quinine about 500 years ago, and this actually had a big impact on malaria control. And so about 100 years ago, they discovered quinine and then chloroquine and some of these new synthetic things. And then they had a big campaign in the 1960s to eliminate malaria. So malaria used to be found throughout the United States. It used to be found throughout Asia, throughout China. And with really concerted use of DDT and treatment and draining the swamps and putting screens in houses, they were able to eliminate malaria from the United States and most of Europe, and I think it's only recently been eliminated from China. But anyway, it disappeared from the United States fairly recently. It remains a big problem in India. They were never able to completely eradicate it. It remains a problem in Brazil and most of South America. I think there's still a few cases in Mexico, but it's for the most part, it's not much around. So we made a big campaign in the 60s. It came back about 30 years later. Everyone had forgotten about it. It came back in a big way. And then people started about 20 years ago saying, oh, well, let's just distribute lots of bed nets and really work on it. And they thought it was going to go away again. And then the last 5 years, it started coming back again. So it went from, I don't know, about almost 1 billion cases a year down to about 200 million. And now it's starting to creep up again. So about 250 million cases a year. And part of this is not just parasite resistance to anti-malarial drugs, but it's also mosquito resistance to insecticides. And so if you look at this is from the World Malaria Report in 2018, we had about, I don't know, 25 countries or so are giving an increase in the number of cases over the past 3 years. And it's probably going to be really bad this year with COVID-19 as resources have been diverted away from malaria control. So we do need better ways of trying to get rid of malaria, and it is possible to eradicate it because it's out of the United States now. We don't have mosquitoes. We don't have people transmitting it. Malaria is not something that we think about. And can we develop sort of, the big thing that consumes me is, can we develop better ways of treating malaria so that, we could possibly think about eliminating it entirely from, places like India and places like, larger parts of sub-Saharan Africa. So that's a lot of what, my laboratory spends its time thinking about. Any other questions at this point? Okay. So let me tell you a little bit about malaria drugs and why the current ones we have aren't so hot. Okay, so most anti-malarial drugs that you'll take are derived from these two natural products. And, we've known that if you drink teas out of these two natural products, you often feel better and reduces the fevers that are associated with malaria. So this has been known in Asia for thousands of years. And then this one came from South America. So this is quinine, or the plant that produces quinine. And it was, they found out, they called it Jesuits tea, and they brought it back. And, whether we had malaria in South America pre-Columbian, I think is a bit of a debate, but I suspect there must have been malaria, which is why people were drinking quinine. So if you've had gin and tonics, this is what gives the tonic the bitter flavor. And this was part of the reason that people used to drink tonic water when they went to India, as a protection against malaria. And the active ingredient is quinine, which is shown here. And then the active ingredient for Artemisia annua is artemisinin. And both of these things basically interfere with the parasite's ability to eat hemoglobin. So when the parasite's replicating in your red cells, as shown right here, they eat up hemoglobin, and then they burst the red cells, and then they eat more hemoglobin. And both of these compounds interfere with that process. And so the parasite can't eat your red cells anymore, and then it eventually dies. Now, the problem with this is that this is only one stage of the parasite, right? So this is just the active replicating form. And these two compounds, as well as derivatives against these compounds, don't affect the other parts of the life cycle. So when parasites are stressed, they form male and female gametocytes, and these are the only stages of the parasite that can survive in the midgut of a mosquito. And so if you're not killing these gametocytes, even though you've taken artemisinin or quinine and you feel better, the gametocytes are still around, so you can still transmit malaria to your neighbor. So to bring up the COVID-19 analogy, let's say you've taken your new drug and your symptoms of COVID have disappeared, but you're still out on the street spreading it, right? So not an ideal situation. And the other thing is that neither of these drugs really act against the liver stage, the quiet stage. And so, we're trying to find completely new types of compounds that can be used to treat malaria. We don't want to make derivatives of these old ones, which is what had been happening for many years. They would take quinine and they'd take the functional group, the aminoquinoline here, and then they'd hook it together. And the problem with this is it didn't address any of these other stages. It's a relatively toxic, half short-lived drug. And furthermore, you have a lot of parasite resistance. So, what we've been thinking about, and I work very closely with the Gates Foundation, is, how do you make completely new versions of anti-malarial drugs? So, I'm going to give two little stories. Hopefully, maybe I'll just give one if we run out of time. One is using important uses for bioinformatics and drug discovery. In the first case, I'll talk about how we developed a new compound using cheminformatics, which is the same as bioinformatics, but you're not going to learn that much about it because it's more related to OChem. If you guys are taking chemistry, it's like, how do you reduce chemical structures into strings and manipulate them? And then I'll talk a little bit about how we use genetics and some genetic bioinformatic algorithms to find new drug targets and drug resistance genes. Any questions at this point? Okay, feel free to interrupt if you want.
So, I guess this is just a question about cheminformatics. I could probably look this up, but anyways, like when you say like just turning a chemical structure into a string, right? Like, would the next step then be just like storing that information, or like actually transforming it to like make, predict product reactions or reaction products?
Mostly just storing that. I'll show you an example of how we use cheminformatics coming up next.
Okay, thank you.
Okay. Anjali?
I just I don't really know how to explain this question. So I've been to India a number of times, and when I was younger, I remember my mother used to have me take like a medicine, I guess, as like prevention. Would those medicines be used, would they be derivatives of those same plants that you were talking about earlier?
Yeah, some may have been. So if you were taking mefloquine, which is used for prevention, it basically that works by keeping sort of a low level. So by the time they go through the liver stage, you still have some hanging around your bloodstream. And so when they go into your bloodstream, it blocks them from replicating. There is one other compound called the atovaquone, which does actually act by a slightly different mechanism of action. And that one also has prophylactic activity. So I was simplifying it a little bit. Probably, I don't know exactly what you were taking, but it was probably one of the two. So it could have been mefloquine, it could have been malarone. But also drug resistance problems. So I was saying what happened in the old days is you get resistance. So chloroquine was super broadly used, and then parasites became massively resistant to chloroquine. And so then they would look at the structure and they would make a new version of it such as piperacin. And a lot of anti-malarial drug discovery was very chemistry driven. So, you know, they would identify the active part of the molecule, so this endyl peroxide bridge in artemisinin, and then they would put it into a synthetic version here, atyrolane and OZ439, and then you'd have a new anti-malarial. But the problem with this is, again, it creates, you know, if you were resistant to the first one, you're probably still resistant to the second one. And then it's basically just Me Too drug discovery. But that was basically what people were doing. So about 15 years ago, and this sort of, it's quite old now. It's getting shocking how long this took. But well, in 2006 and 2007, I was involved in a project to take phenotypic screening to malaria. And at this time, it was actually considered quite radical, but I was working at the Genomics Institute of the Novartis Research Foundation, which is here in San Diego, close to the UCSD campus. And we had been working with a lot of people, including some engineers from the automobile industry that had been building robots. And this is one of the robots that's shown. And we'd also been assembling lots of chemical libraries and putting them into towers. So does everyone know what a chemical library is? So the term library is used a lot in biology and chemistry. And it means it's a collection of compounds that was sort of randomly assembled. And it's called a library because in most cases, you know exactly where it sits in a stack. So with a library of books, you know if you take a code, you know that it's on the 4th floor in, you know, stack 36 on, you know, on the 4th row. And so, shh, kitty. And so a chemical or a biological library is the same thing. You have large collections of compounds, lots and lots of random compounds, in this case, millions of compounds. And we know the location of each one, and they're arranged in these 1536 well plates or 384 well plates. And what the robots were able to do is to take tiny amounts, you know, like basically the size of a piece of the size of a head of a pin, and then to take them out one at a time and then to put them in these plates. So we said, well, we have these libraries that can these robots and these libraries and can dispense it. Let's just see, you know, if we want to make new anti-malarial drugs, let's just put the chemicals and the parasites together and then see if we can find ones that kill malaria. And doesn't seem that complicated, but we needed a lot of technological advances in order to get this whole thing to work. So we took a bunch of chemical compounds, and then we dispensed them into plates, about 5 microliters. And then we added malaria parasites, which grow in red cells. And then we let them sit together for 2 days. And then we added some DNA binding dye that measured the growth of the malaria parasite after 48 hours or 72 hours. And so we were able to go through 2 million different compounds, and we knew what exactly was in each compound, in each well, and we knew the chemical structure of what was in the well. And then we just tried to optimize this so that we could really get nice signal to noise, and we could figure out whether or not the chemical that you put in had killed your parasite. And we put these in at physiologically relevant concentrations, about the same concentration that you would, if you were to take aspirin, aspirin would accumulate in your bloodstream, and it would be in the low micromolar range. So we actually ran a screen of about 2 million compounds, and we were able to identify these hits. This is, I actually just stole this from Wikipedia, but this is what our plates would have looked like. This red spot would be a place, where your parasites weren't growing very well. And then the other ones would be places that they were growing fine. They didn't care about the compound. And then would have controls over here, something that you knew killed parasites, or you would have some wells that had no parasites in them. So, and then you process all of this data, and then you go, oh, I found 8,000 spots where there looked like there was no parasite growth. So the next thing you do, and this is how you run a screen, is you go back and you type all of this into your computer program, and then the robots go back and then go find the wells, the source plates for the compound, and find the compound that you're interested in, and then they redispense it out in dose response. Okay, any questions at this point? Gina?
Oh. So if these drugs are potential things that the patient could like take, then wouldn't it be different from just exposing the parasites to the drugs outside of the human body versus actually them interacting inside?
Well, it turns out that it's actually very similar. These aren't exactly drugs you could take at this point. So these are chemical starting points. So, so there's a difference between something that can kill malaria in a Petri dish and a drug that you can actually take. And the biggest difference between the two, between the compounds that we have here and a drug that you can actually take, is that the chemist will make modifications so that it improves its stability in your body. So let's say you drink a concoction that has quinine in it, and most likely your body will detoxify the quinine very quickly, and therefore the concentration of that compound in your bloodstream and its ability to kill the parasite will go down pretty rapidly. And therefore, you won't be able to really get a cure when you actually were to take it. And then the other problem, of course, is that with these random compounds that we have here, some of them are toxic, right? And some of them you'd never want to take. So how you go from these, what we call screening hits, to a drug is the whole area of drug discovery. Does that answer your question? Okay. Oh, David, do you have another question?
Yeah. So you mentioned that some of the drugs were, well, I guess I have a couple questions, but I'll start with this one.
I perhaps shouldn't have called them drugs. Okay, they're chemical compounds that are drug-like.
Oh, okay. Yeah, but so you start with some chemical compounds that could be potentially highly toxic. How do you figure out that they're toxic? Is it something that's predicted pragmatically?
Yeah, that's actually kind of hard to do. A lot of it is done empirically. And so we have run these screens over and over again, and we accumulate data for each one. And therefore, one way to look at the toxicity is to say, oh, this compound, it killed malaria, but it also killed, these breast cancer cells, and it also killed E. coli, and it also killed, these cells, and it killed everything. So it probably is toxic. Now, the compounds that we ended up working on often were very specific for malaria. So they killed malaria parasites, but they had really no effect on other types of human cells. But again, we had 2 million compounds, so this becomes a big data informatic exercise. So you can do it informatically, but another way is that ultimately you perform a series of tests in the drug development to make sure that there isn't much toxicity. And if you ever take a class in drug development, I mean, some of you may end up in industry, and making drugs is a big part of the biological industry. And, late stage toxicity testing involves taking a couple of animal models and then giving them really, really high concentrations and finding out what's the concentration that makes them sick. So looking for a lot of different evidence of toxicity, like enlarged hearts or enlarged spleens or behavior changes or loss of weight, things like that.
Yeah.
Okay. So the next thing you do, you've identified about 8,000 compounds, and the next thing you really want to do is quantify how much effect they have on your parasites. And so then you go back, you have computer programs that go back, fish out the compound, and now what you do is you dispense it out in reducing concentrations. So you start at 10 micromolar, 3 micromolar, 1 micromolar, 300 nanomolar, and then you basically do what we call a dose response curve. And then you put parasites back in, and what you want to see is a dose-dependent decrease in parasite growth. And then based on exactly the cutoff value, the 50%, the level that causes 50% reduction in the amount of parasites, you can define what we call an IC50 or an EC50, which is the inhibitory concentration or the effective concentration that gives you a 50% reduction in your response. And that really tells you how potent your compound is. So we found a bunch of new stuff, and then we also had sprinkled in known antimalarials in our screen. And so we knew that it was working pretty well because we rediscovered a lot of known antimalarials with our chemical screen. So here, again, this would have been done for 8,000 compounds individually. You plot the response, the amount of parasite growth that you have is a function of drug concentration. You compute a curve, and then you do a curve fitting algorithm to figure out the 50% mark. And then that tells you what, quantifies how good it was. And if you ever study cheminformatics, we put all of the data into the public domain. And the idea was that we can't use all of the data. So we put the chemical structures and the IC50 values and toxicity measurements all into the public domain, into the Chembles. So have you guys heard of the NCBI databases where they store sequencing data? So this is similar, except we're sharing chemical data. And there's a big database called Chemble, and there's also PubChem, which is the American version. And this will have a lot of chemical structures and inhibitory values, and it's the other end of bioinformatics, which is cheminformatics. And I'll show some examples now. So these are what chemical compounds look like. And you've probably seen some of these in your classes and so forth, and you've probably had to draw ATP and things like that. How many people know the structure of ATP? Well, I can't, but if you've taken biochemistry, you probably know the structure of ATP, and a lot of compounds that are drug-like are very similar to this sort of cofactors like ATP, except, so they're meant to confuse your enzyme, right? The enzyme wants to bind ATP, but it can't find ATP, it finds a drug instead, and the drug gums up the enzyme, and then the enzyme can no longer function. And this is the basis for a lot of pharmacology for drugs that you take. And for example, with cancer medicines, a lot of them would be ATP mimics. So let me just show you one of the basic things that happens with cheminformatics. So we did a huge screen with our library, and we got lots of things out that looked like this. And we knew the structures of the compounds in our libraries because people had used NMR and other sorts of methods to decipher their chemical structures. And we were able to, so we could get the structure, which is shown here, and then we computed these IC50 values. So the IC50 for this compound was 305 nanomolar. The IC50 for this compound was 480 nanomolar. So how do you organize all of this data? So we have 8,000, entities like this, and we have an IC50 and we have a structure. And the next question is, how do you parse through this data? Well, one way is to just take the most potent compounds, and that's kind of sort of the boring way of drug discovery. But the more sophisticated way is to take all of this data together and say, what structures do we see that are popping up over and over again that are different than known drugs? Because we're really looking for something new. So the algorithm that people use is basically by breaking your chemical structures down into what we call fingerprints. And so this is a Tanimoto, actually I should have written that out. This is, oh yeah, here it is. It's a Tanimoto coefficient. And basically, you generate fingerprints or hash functions for your different compounds. So let's say you're comparing this compound and this compound, and you break down all of the different entities within the compound, and you give it a 1-1 if they both have it, and then you give it a 1-0 if one of them has it and the other one doesn't. So then you create fingerprints, which are shown here, which tells you the similarity between compound A and compound B. So for this example, the number of bit sets in both of these two compounds equals 3. That would be 1, 2, 3. And then the number of bit sets in 1 but not in 2 equals 2. The number of bit sets in 2 but not in 1 equals 0. And the Tanimoto coefficient is A divided by A plus B plus C. And so this would give you a 60%, the Tanimoto coefficient comparing this to this would be 60%. So you can then construct matrices out of this and then use this to actually create hierarchical trees where you can now group your compounds based on how similar they are to one another. Questions? And I didn't really go into the algorithm that you would use to create hierarchical trees, but there's a lot of them out there, and it's basically the same algorithm that you would use in population genetics to arrange pieces of DNA by similarity. Alex, do you have a question?
Yeah, I had a quick question on what would you normally use? I guess what programming language would you normally use for the clustering?
Well, there's all sorts of one of the tools that you can use is R. Have you guys heard about R?
Yeah, I'm working with H cluster right now in R. That's why I'm curious.
Yeah. So there's a set of programs called ChemMineR that are devoted to cheminformatics, and basically they use the R programming language. So ChemMineR can be used, and then there's also a variety of more proprietary softwares that are associated with commercial packages for clustering. This gets into sort of more hardcore algorithms, which is not my strength, frankly. But I mean, I know how to use ChemMineR. I do a little bit in my laboratory and things like that.
Okay, thank you.
Okay, so this is showing the clustering. David, you raised your hand.
Yeah, sorry. Just a quick question. I was wondering if there's like more information that might be important too, like when you're doing this.
Yeah, actually, that's a very good point. So this is used to group compounds based on similarity. But the other question you might want to have is what is known in the literature about this compound, and they basically will use the same algorithm to do a similarity search. So let's say we take this compound. Actually, if you want, if you go to the web, I can show you exactly what we would do with that. So we can take one of these compounds, and this is a two-dimensional object, but I can convert it in a program that we have to a string. So if I take this two-dimensional structure, I can convert it to a string, and then I can paste this string into a search algorithm that runs a little bit like Google for chemistry, and then it will run the Tanimoto search on all of the chemical space out there, and then it will come back and tell me what's similar and what has been used. Okay, let me show you an example of this. Okay, I'm going to share my screen again. Oh, here it is. Okay, right on top. Okay, this is one of the compounds that we screen. I've got to go open. I'm assuming you can see my screen. So I'm going, this is the compound that we discovered. So it's now called ganaplacide. And here is the structure here. And if we go down, there's a lot of different ways to call it, but one of the ones that you can call is called the SMILES code. So here is the two-dimensional structure, which you're sort of familiar with from chemistry class. It's really hard to paste a structure into a Google search document. Is that right? I mean, you can't take a picture of your friend and put it into Google and find out everyone that looks like him, right? So likewise, you can't take a chemical structure and paste it into Google and find everything that's similar. So what we do is we convert it to what's called SMILES, and this is a way of converting this 2D object into a 1D object. Do you guys see that SMILES code here? Well, tell me if you can't. Okay, yes, good. I like to see the thumbs up here. So I'm going to cut and paste this. So I'm going to copy it. And I'm going to go over to another program that we use called ChemDraw. And there's a free license for ChemDraw in, if you guys are taking OChem, you might have to use it. Has anyone seen ChemDraw before? Anyway, we have a free license for ChemDraw at UCSD. And if you ever have to do any OChem and need to draw any structures, this is what you would use. So, I copied this string, this CF, basically a two-dimensional string, which I'll show you right here. Oops. So that's the structure. And, oops, well. That's the SMILES string that I copied, and then I can use paste special and paste from SMILES. So ChemDraw will interpret the SMILES and give me something like this. So how do you do this search? So this is a way that the SMILES string, which is a way of representing chemicals, is a way to talk to a search engine. So I'm going to go back here, and I'm going to go to PubChem. Okay, do you see my PubChem window? Okay, great. Okay, so now I'm going to paste in my SMILES code, and I'm going to search. And look, it found ganaplacide. See that? So you can then use the similarity coefficient to find everything in the PubChem database, which is similar based on the algorithm that I just showed you. So now we can go find similar structures, because I want to find out if I want to write a patent about this, that if someone else is working on, that's basically what people want to do. And this is another compound that I discovered or worked on, and it's very closely related. It's differing. It has a fluorine base instead of a chlorine, basically, but it's very, very similar to the two. Again, you would see a fluorine here instead of a chlorine. So the PubChem search engine was able to use an algorithm and quickly look through the search of billions of, or I don't know how many entries you have in PubChem, but it's definitely probably about 100 million. It was able to search through all of that in just in a split second and then return this, which I think is quite remarkable. Any questions? Has anyone ever heard of PubChem, even in your chemistry classes? Yes? Because we didn't have PubChem when I took chemistry. We just had, you know, beakers, and we isolated caffeine from tea, and there was absolutely no informatics whatsoever associated with chemistry when I took chemistry. So I don't know what they're teaching you these days, but chem informatics and these ability to process these types of structures is very important to, you know, the process of drug discovery. So let's see. I'm going to go back to my PowerPoint, if that's okay. Okay. Okay, so I'm just going to speed along here. I'm not going to go into that. So anyway, the way that we got to Ganopoliside is we arranged all of these based on similarity. So we used a clustering method and then the similarity searching algorithm to basically bin the different compounds in similarity. And one of the things that we noticed that there were 3 copies of this compound here in the library. And this was different based on the chemists looking at it than most of the known anti-malarial drugs. And yet we found 3 different versions of it within the library, all of which were fairly potent. And this was at a rate that was higher than expected by chance. So we also knew everything that was in the library. And the fact that you had 3 copies of this type of molecule in the library of 1 million compounds, and all 3 of them were active, the probability of that happening by chance was not particularly good. So you can assign a p-value, but that doesn't tell you whether it's really attractive. But that was one of the reasons that we decided to work on this. So the next part that came along was some hardcore chemistry. That's just the algorithm. And what we then, now this is now stuff that I wasn't doing. This was work by Arnab and Adi, and so I was sort of screening informatics. And what we did is we took this chemical structure here, and then the chemists started making modifications to it. So they used their chemistry skills to change this from a fluorine to a chlorine, or they added different decorative groups here, or they took away this aryl ring here, or they made a variety of different modifications. And so basically, when you would synthesize this compound, they would put in different tweaks that would result in you getting different things out. So this is really the way that the medicinal chemistry worked. And every time they made a new version of this, we would test it in the parasite assay that I showed you before. And we were looking for things that would increase the potency against malaria while decreasing the toxicity. And we were also testing to see how long the compound would stay in your bloodstream, and looking for things that would stay around in your bloodstream for at least 6 hours. And furthermore, we wanted to see things that you could take as a pill. And we were looking for cheap pills, actually, because malaria is not for people that can afford $1,000 medicines. Or $1,000 medicines are not for people that have malaria. So we made, ultimately the chemists made about 1,000 different derivatives over about 1.5 years period, all of which were tested in these very high throughput assays. And we ended up with a compound called CAF156. And this thing was actually able to cure mice of malaria. So we would give mice orally a dose of it, and oops. And so if you give mouse malaria and you let them go, they're dead. All of them are dead in 6 days. And if you were to give them either GNF179 or CAF156, the compounds that I mentioned before, very similar to one another, you gave them a single dose of 100 mg per kg, which is not a big pill. Their survival went out to 20 days. Which is, and then so obviously if you give them multiple doses, you could increase the survival even more. And of course, you also have the immune function that kicks in. And furthermore, if you give these mice a dose of this and then you let the mosquitoes feed on them, it actually protects them entirely from malaria as well. So pre-treating the mice with a 15 mg per kg dose of GNF179 completely prevented the mice from actually getting malaria. And furthermore, we discovered that this compound kills gametocytes as well. So if you take GNF179 or CAF156, they're very similar to one another, it actually prevents you from spreading malaria to your neighbor. So it blocks transmission, and furthermore, it's relatively simple to synthesize, and it has good pharmacokinetic properties. So this had all the things that were suitable for actually going into human trials. So the next thing was to go and to do toxicity testing. So first they did toxicity testing in animals, and it continued to look like it was non-toxic. And the next thing was to go into phase 1 clinical trials in humans. So here they take the lowest, they take the dose that gave no adverse events in animals. So they put in a whole bunch of things and figured out what made it sick, and then they backed it down below that, and then they got human volunteers that would escalate up to that dose. And basically the human volunteers this was a study that was going in Australia. They're doing the same sorts of tests, with COVID-19 drugs right now. So so this has been through extensive testing in animals, and now you get human volunteers that take, basically a very, very low dose and then a slightly bigger dose, and they take it in a hospital, and then the doctors all look at you and make sure that there's absolutely nothing going wrong. And then they figure out, what sort of a safe dose would be to give it to humans, and then they take it off to the field and they test it with people that actually have malaria. And it actually turns out that ganaplaside does actually cure malaria. So this is now going into phase 2B and trials. It's being partnered with a drug called lumefantrine, and now it's being extensively tested throughout Africa. And there's a good chance that this drug will actually be licensed. So that's how we used bio and cheminformatics to find a new drug for malaria. And I'm not sure how much more time, but maybe we should just take a few more questions at this point. And this is probably, you'll have other people telling you more about genetics of malaria or genetic systems. So I think hopefully you've learned something new here. Questions?
I have one question. So have you used any sort of like unsupervised ways to synthesize not synthesize, but like discover the compound that could be used in malaria treatment, or is it all like based on the algorithm and look for the similarity?
Well, for this case, we tended to use the algorithm and looking for similarity for this one. In another case, I've actually been involved in putting 2 compounds into clinical trials. In the other case, it was just, oh, this is the most potent thing we found. And so we worked on that one for a while. But the compounds that come out of these screening libraries are usually not suitable for testing in people, because you got to make them less toxic. You got to make them more potent. You can't give someone, so if you go in for like cancer treatment, someone may put an IV drip directly on your arm, and you can get huge doses of cancer treatment, but no one's going to give you an IV drip for malaria, right? these are kids in the village, and they're going to get a pill, or they're going to get a suspension. They're going to get aspirin-like compounds. And so you have to get it potent enough that you can give it as a pill, because no one's, you can't give a pill that's the size of a quarter, right? No one could swallow it. And so so you got to get the concentration, or the ability to kill the parasite down to a potency where you can actually give it as a pill, which is something we don't really think about. But, when you're learning about molarity in chemistry, molarity affects how much compound you have in your bloodstream, and that affects how much you actually have to take to get a cure. So anyway, those were compounds that came from phenotypic screening. So we tested compounds' ability to kill parasites, and you've also probably heard about structure-guided drug discovery. And someone's probably come and told you that you have a nice target, and then you can do docking. Have you guys heard of docking? And
Yeah.
Well, that's another way of kind of going at it. Where you say, well, here's a crystal structure, and I'm trying to fit a compound in it, and it's very rational. But we didn't have a good target, so we just took this brute force approach, and it actually worked. So, and you probably aren't hearing much about this, but phenotypic screening is a very good way of sort of bootstrapping your way up when you don't know what a good target would be. See, if we had gone for target-based drug discovery, we would have probably been working on dihydrofolate reductase, which is one of the known targets, except that there's this huge resistance problem with dihydrofolate reductase, so it's probably not the best target. And if we were having another hour, I would tell you how we figure out how these compounds are working. I'm not sure how much time we had scheduled. Was this just for an hour?
Yeah, it was scheduled from 5 to 6.
Yeah. So it looks like we're 6 o'clock now, and you guys have more to do. So if you want to have me back, I'll tell you how we find targets. But that's basically what we're doing. Some of the informatics that we use in my laboratory.
Okay. So if nobody else has any questions, then I guess I can end the meeting. Thank you for us, Dr. Winzeler.
Yep.
Thank you. This is my first one of these. It's been really interesting.
Yeah.
Okay.
Well, email me if you have any other questions that come up.
Thank
you. Of course.
Yep.
Bye.
Bye.